इस मॉड्यूल में हम बात करेंगे एंजाइमेटिक सीक्वेंसिंग के हवाले से यूजिंग सीक्वेनेज एंजाइम. Sequenase is a modified version of a bacteriophage T7 DNA polymerase. Bacteriophage basically is a virus that infects the bacteria. So this enzyme sequenase comes from a virus which is bacteriophage T7. This enzyme and the cleanove enzyme of E. coli, which is a component of DNA polymerase 1 of E. coli, the bacterial enzyme as well as this is the viral enzyme, the sequenase, they have no activity 3 to 5 prime exonuclease activity, which is present in regular normal DNA polymerase 1. So use of sequenase or use of cleanove fragment or tag polymerase, this is choice of the researcher and the experimental condition that which enzyme should be preferred over which enzyme at what time and under what conditions. अब हमारे पास सीक्वेनेज के कई वर्जन्स मौजूद हैं. One after the other modification because of researchers, they were using this enzyme, they were adding modification and modification. The best result comes from by the use of sequenase version 2, and the reaction or the enzymatic reaction is carried out as stated under. Like इससे पहले हमने बात की denaturation would be the first step of double stranded DNA template. अगर single stranded है denaturation की ज़रूरत नहीं है. अगर template double stranded है we need denaturation. The second step would be annealing of the primer to its complementary sequences, and the third step would be the brief polymerization reaction. अब ये brief polymerization reaction जो है that is basically the step when we are going to label the primer, because during this brief polymerization reaction in which we add 20 to 100 nucleotides, we basically use one radiolabeled nucleotide which is normally DATP, but it can be any of the radiolabeled nucleotide. Now this radiolabeled nucleotide gets incorporated at different places in that brief polymerization step, and it acts as the labeled end of the newly synthesized strand. Once it happens, the brief polymerization step is over, then we proceed towards the extension and termination reactions, and extension and termination reactions are going to be performed in 4 different tubes which are labeled as A tube, G tube, T tube and C tube. All these tubes contain all the ingredients components similar except the dideoxynucleotides. Each tube is going to be provided with only one type of such nucleotide, dideoxy, which is going to be responsible for the termination of the strand synthesis at that particular nucleotide. Sequenase version 2, it gives us best results with single-stranded DNA template as well as double-stranded DNA template if this is denatured before proceeding further.